board. So yeah, so that we don't miss anything. Okay, Sophie did a very good start, and thanks Vishal and Gary for your time and mentoring. I think both of you did a terrific job. And our next presenter is Zyaire Beasley. And unfortunately, due to clinical obligation, Kiran and Jay cannot be here. So I'm gonna do the introduction for Zyaire. Zyaire C. Beasley is from Propel Andrew Street High School in Munhall. And during the initial interactions, Zyaire expressed her interest in pursuing a medical career. Accordingly, she was involved in the data preparation part of the artificial intelligence-based choroid biomarker detection project, hoping that it will help her familiarize herself with ophthalmology. In particular, she was trained to build ground truth data required to validate AI models developed in-house. She got familiar with the posterior segment part of ophthalmology, OCT imaging, and tools such as Imagej software. We wish her all the best and hope the learning here will help her future endeavors. And this is the introduction written by Kiran, her mentor. And Zyaire has been participating in the roundtable, so we have met a couple of times also. So Zyaire, can you share your screen?
It's not giving me an option to share my screen.
So can you try the green button that says share content?
No, I don't have a green button that says share content.
I think I have enabled the sharing functionality. So there should be if you are using a computer, there should be a green button on the bottom bar of your Zoom.
I'm on a MacBook and I don't see.
Wait a sec. Have you
sometimes?
Yeah.
Sometimes if you move your finger on the book, or you move your mouse, it'll pop up on the bottom. Try it. Maybe give that a try.
No, it's still not popping up. I don't.
Wait a sec. So Zyaire, can you try sending me your PPT so that I can share it for you?
Yes, I can share it with you.
Sending it to CHENY1 at pitt.edu.
Right, you said
I said
CHEN.
CHEN. Y. Number one. At pitt.edu.
At pitt.edu. Okay.
Yeah. Okay, I've seen it. So is that a PDF file? Oh no, that's not you. Oh, here you go. Did you send it, Zyaire?
Yeah, I shared it. Are you able to see it?
Not yet. Okay, let me see. Yeah, there seems an email delay. So let's double check again. If you move around your mouse, can you see a bar of menu on the bottom of your Zoom?
No, I can't at all. It's on in the left bottom hand. It says it has the audio button and the camera button. I can try to see if it works if I join on off my phone instead.
So do you have a bar saying security participants chat?
Security participants chat.
Yeah, and share screen and polling.
No, I do not. It no. I don't see that.
So how about how about let's do that. Let's move you to the next speaker. And then when I receive your email, maybe I can bring you back for your presentation. Can we do that?
Okay, so you just want to see if it works, if you're able to see it?
I still didn't receive your email yet. So can you see, I remember we contacted yesterday. Can you find the email record we did yesterday and reply to that email and attach again to that email so that you can contact me again with that email?
Okay.
Just in case if there are any typo for the email address. So Zyaire, I'm gonna put you to the next speaker after following the next speaker so that we don't be so delayed on time.
Okay.
Okay, all right. Send me another reply to the previous email so that I can receive your attachment. All right,
Okay.
let's move on and then until Zyaire figure out, we can put her back in for her presentation. But our next speaker is Yingwen Lin, also Venice. Mentor is Dr. Kun-Che Chang. And so Venice, Kun-Che, can you do a brief intro?
Yes. Hi, everyone. I'm Kun-Che Chang. I'm the assistant professor of ophthalmology at the University of Pittsburgh. First, I would like to thank this Hillman Foundation for giving us this opportunity to train high school students. And I also want to thank Yingwen for co-organizing this. I also want to introduce our summer intern this year, Venice. She is a rising senior at North Allegheny Senior High School, and she has a strong interest in pursuing a medical related career, and I can see a bright future ahead of her. She's an amazing student and been very productive and helpful on several projects in my lab this summer. And I'd love to have her around the lab for longer and welcome her coming back anytime. Today, she will give us a presentation about her study. Now, let's welcome Venice.
Can you guys see my screen?
Yes.
All right. Hey everyone, I'm Venice from Dr. Chang's lab. So my presentation today will be centered around therapeutic strategies for ocular diseases. Instead of doing one big project, we decided to do 3 smaller projects. So let's jump in. This is a brief overview of our project 1, which is the influence of ascorbic acid on hyperglycemia-induced cell growth inhibition. Diabetes is a rapidly growing disease today that is affecting many people because of many reasons like sedentary lifestyles and family history. A common symptom of diabetes is hyperglycemia, or more commonly known as high blood sugar. If hyperglycemia is left untreated, then it will start to induce oxidative stress on different parts of the bodies, including the eyes, which can cause a condition called diabetic retinopathy and blindness in the long run. The purpose of our experiment is to find out if we treat hyperglycemia with an antioxidative agent, which in our case, we're using vitamin C, will it protect the eyes against hyperglycemia-induced abnormality. Firstly, we're going to talk about the experimental design of the project. We decided to use the ARP19 cells, which are the outermost layer of the retina, as you can see over here on the bottom right. It functions as a light absorption and interacts with photoreceptors. It is a very important part of the eyes because if it's attacked by hyperglycemia, then it can also cause photoreceptor death as well. So the big question today is how can we protect RPE in diabetic eyes? After deciding on a test subject, we turned to designing how they will be treated. So we had 10 96-well plates, 5 plates of figure 1, which is treating them with low glucose and high glucose for 24 and 48 hours, and 5 plates of figure 2, which is treating them with low glucose and high glucose for 48 hours, but half of them have 100 micromolars of ascorbic acid or vitamin C added to them. After the culturing of the cells, we ran the samples through MTT. The MTT assay is a method for measuring the cell viability. As we can see here on figure 1, we used low glucose as our control and set it to 100% viability and compared it to the high glucose treatments. We can tell that the cell viability is lower for high glucose compared to low glucose because high glucose for 24 hours is roughly around 75%, and for 48 hours, it's at 70%. Now, if we look at figure 2, which is the ARP19 cells that are exposed to high glucose but treated with vitamin C, they have a significantly higher cell viability than the control group, meaning that vitamin C can rescue the cells from hyperglycemia. Next, we have the Western blot, which determines if the cell viability reduction is from apoptosis. I'm going to give a little rundown of the procedures of the Western as well. First, we collect the samples from the cell culture after treatment and then ran it through the Western blot, then probed the blot with PARP, which is an apoptotic marker. After imaging and analyzing, we found that the data does suggest that hyperglycemia-induced cell growth inhibition is mandated by apoptosis. If we look at the graph on the bottom right, the higher the ratio, the higher the cell death. If we set low glucose as 1, high glucose would have almost 50% more cell death. Finally, we can conclude that hyperglycemia-induced cell growth reduction can be prevented by the intervention of ascorbic acid or vitamin C. As for future works, we can investigate a broader spectrum of similar hyperglycemia-induced stress in other organs and the effect of ascorbic acid on them. and discovered the different ways to incorporate ascorbic acid in animal studies to prevent hypoglycemia-induced damage and examine the underlying mechanism of high glucose-induced stress or oxidative stress. Okay, so now we're on project 2, which is focused on suppression of microglia-induced inflammatory responses. Microglia is a type of glial cells that function mainly as an immune defense system. When they're activated, they will secrete cytokine. In our case, we're looking for the TNF-alpha. If there is excessive cytokine secretion, then it can cause damage to good cells and trigger inflammatory responses. So what we're trying to figure out is to determine if sorbinol can suppress the cytokine secretion to be used as a simulation of activated microglia. We're using LPS, which can activate the microglia to secrete more cytokine. So the picture on the left is trying to show you where the microglia is located in the retina. So they are the green specks where the arrows are pointing to, and the purple are the RGC. Moving on, the ocular inflammation can cause vision loss like uveitis. As we can see here on the bottom is a comparison of a normal eye and an inflamed eye. We're using sorbinol, which is an autoreductase inhibitor that could be a therapeutic agent to prevent LPS-induced microglia activation by reducing the TNF-alpha cytokine secretion. So we ran it through a ELISA test. If you look at it, microglia was treated with different concentrations of LPS, and we found out that microglia that was treated with 100 nanograms per milliliters of LPS secreted the most TNF-alpha. So if we look at the graph on the right, we can see that LPS-induced secretion was attenuated by sorbinol treatment compared to the DMSO, which is our control. In conclusion, we can say that sorbinol can alleviate ocular inflammatory responses. Finally, we have our project 3, the stem cell differentiation. This is one of our longer projects that the entire lab is focusing on at the moment. I was very lucky to have the opportunity to help maintain 2 of our stem lines. So the pictures here are from my oldest line. We're basically trying to differentiate human embryonic stem cells into retinal organoids. Which you can see here are the embryonic bodies, neuroepithelium stage, organoid protrusions, and retinal organoids. And the picture on the bottom right, or like the last picture, is what we want to see because it is illuminating red, or it has TD tomato signals, which means that it is a RGC-like cell. Lastly, we have some immunostains that we did on the organoids. On the left is an image of the cross-sectioned organoids that are stained with DAPI and RFP. So the blue from the DAPI represents all of the nuclei, and the red from the RFP means that they are RGC-like cells, which is what we want to see. On the bottom right is a stained dissociated organoid. Similar to the cross-section, we want to see the burn-3A TD tomato signals, or the RGC-like cells, which makes up actually 73% of the total cells. Our long-term goal is to transplant the differentiated stem cells into RGC lost eyes in glaucoma and optic neuropathies. All right, that's my presentation. I feel really lucky to join the Chang lab and have the opportunity to do everything that I presented today. But before we end today, I really want to thank everyone in my lab, Dr. Chang, Chandler, Liz, Suresh, and Huichun, the University of Pittsburgh, and the Department of Ophthalmology. A special thanks to Dr. David Boone Solomon and the Department of Biomedical Informatics and the Hillman Cancer Center. And lastly, to our funding sources. And a big thank you to everyone that came to my presentation today. I really appreciate it. And if you have any questions, it's the perfect time for that.
Again, another great talk So Venice, can you share with us your face and select one of your favorite slide? And Kun-Che, can you help me again doing the screenshot?
I have a question, and it's not so much about the presentation, although it was very impressive. Great job. Did you draw the picture on the final slide?
But yeah, I did.
That was great. That was a nice way to end. Very nice.
Thank you.
I have another question, not related to the wonderful presentation, but are you looking at, have you looked at where you're going to go to school or
For undergraduates?
colleges? Yeah.
Yeah. I've been looking at several schools. I
Is
really
Pitt
want
one
to
of
go
them?
to, yeah. Pitt is one of them.
Okay,
Like, my top choices is Pitt is one of them.
good.
Dr. Chen, I just sent the screenshot again to you.
Any more questions to Venice? No more. Let's sorry for the technical difficulty for Zyaire, and now I have her presentation, and I think we can put her back for her presentation, and I am gonna share content. I don't know how to do it myself. Why screen? The screen. Okay, now you can see my screen. Here you go. Let's go to the first slide. You present. Okay, Zyaire, it's yours.
Okay, so my name is Zyaire Beasley, and for my project, we took ground truth data to validate artificial intelligence models developed to quantify the choroid layer of the eye using OCT images. And my mentors were Dr. Kiran and Dr. Chhaablani, Department of Ophthalmology, University of Pittsburgh School of Medicine. Can you go to the next slide, please? Okay, so the choroid is the vascular layer of the eye, lying both between the sclera and retina. And studies have shown that choroidal structural changes are associated with many diseases, including age-related macular disease, central serous retinopathy, and pachychoroid. Choroidal dysfunction is implicated causally in several posterior segment eye diseases, so there is considerable interest in characterizing choroidal structure in terms of specific biomarkers using in vivo imaging. Can you go to the next slide, please? So the clinical studies that have been used so far have been based on optical coherence tomography, also known as OCT images. Optical coherence tomography is the imaging technique used to capture micrometer resolution 2 and 3-dimensional images from within biological tissue. This was used to detect changes in the retina and identify disease subtypes. Next slide, please. Okay, so the OCTB scans are then ran through the artificial intelligence model. This is a model based on a collection of artificial neurons that loosely model them in a biological brain. The training and data are the OCTB scans, and the output images of the OCTB scans after being ran through the AI model are the predictions of the diseases. Now, the Imagej procedure is very, very important, and it is a number of steps for marking the boundaries of the chloride layer. And this is important because you want an accurate screening of diseases. Next slide, please. Okay, so our main goal with this was to achieve automated chloride segmentation and wide field OCTB scans. We needed to make sure that the chloride was detectable in the OCT images so that it's marked and presented correctly in the output images. Next slide, please. Okay, and these are the steps for the Imagej procedure. All right, first you have your OCTB scan, and then next you have the first chloride inner boundary marking by using the segmented line tool. Then you smooth the chloride inner boundary marking with the fit spline tool. After that, you have your final chloride inner boundary marking. Then you repeat the same steps with the chloride outer boundary. And finally, the boundaries of the chloride are marked so that it is detectable in the output images. Next slide, please. Okay, so finally for our results. So overall, towards the left are the initial OCTB scans, and this is our artificial intelligence model. And following the OTB scans, these are, I'm sorry, did someone say something? Okay, all right, well, all the way to the right shows Imagej procedures, and these are the output images of the chloride after going through the AI model. And 20 of 100 images were marked in all. Next slide, please. And for our conclusions, we had successfully created ground truth data of 20 images to facilitate automated chloride layer segmentation using OCT images. And for our future plans, we plan to add even more images to the ground truth data. Next slide, please. And I would like to give many thanks to Dr. Kiran for it was very well working with him on this project, Dr. Chhaablani, Dr. Boone, Solomon Lipschitz, Dr. Chang, and the Hillman Cancer Research Academy. And if there's any questions, then that will be the time.
I have a question, Zyaire. Nice to meet you. Before you joined this research team, did you know a lot about artificial intelligence or anything, or what did you, or is this completely new for you?
This is actually completely new. I did not know anything about it, so it's definitely been a learning experience.
It's a really complex concept, so this is really wonderful. It's a lot of information to take in just a short amount of time, so great job.
Thank you so much.
Do you think this will get you interested in the computer aspects and the informatics aspects of your projects in the future?
I'm sorry, can you repeat the first part?
Do you think this might interest you more in the computer processing aspects of your projects down the road in your future?
I think it will, because I didn't know that there was so much behind it all. Like there's so much, like there's so many procedures and data and things that you need to get in order. So I think it will, because I'd like to look into it more now that I know that there is so much more behind it all.
Good.
Okay.
So Zyaire, please show up your face and let me know which slide you want to show. with your
Slide
face
7.
and we will get a screenshot for you.
Right.
Maybe this
Can
one?
you guys see me?
Yes.
Yes.
Okay, let me put it on screenshot.
I can't see myself.
Oh, and
I don't know if you guys-
thanks, Craig and Kun-Che. I got your screenshots. Can you do it again for me?
Yep.
Because yeah,
Yep, no
thanks so
problem.
much. And Zyaire, show up your face.
Yep, we're good. I got her.
Okay, good,
Oh, you guys got me?
Yep, thank you.
great. Okay, our next speaker is Marcus Waller from Dr. Kip Kinchington's lab. And I'm gonna stop sharing myself. And now it's yours.
Okay. So let me introduce Marcus. Marcus has been in my lab for the last 6 weeks, and he's a junior to senior transition between at the Pittsburgh Science and Technology Academy. In fact, I'd heard a little bit about Marcus from my wife, who's a teacher there as well. So Marcus chose to do a good hands-on lab work project. So he came into the lab every day and really got involved in the heavy-duty bench work of some solid projects that we're doing, and most of it's molecular virology, but I think he enjoyed it and he learned a tremendous amount. And I have to thank Michael, my lab manager, who guided Marcus with the hands-on bench work. But I think Marcus really enjoyed the lab, right, Marcus? Yeah, so we had a great time in the lab, and Marcus is one of those people, he's an explorer. He might go into education or he may go into medical school. I think hopefully we've swayed him towards a scientific interest in his career. So Marcus, take it away. Tell him what you've been doing.
Yeah, thank you for the introduction. So my project this summer, with the help of Dr. Kinchington and Michael Yee, was generating deletions of the latency-associated RNA of Bacillus-Zoster virus by recombinating. So Bacillus-Zoster virus, or commonly called VZV, causes chickenpox during its initial infection. You can see chickenpox lesions on this side of the image, and they can then cause a latent infection. So they go down the nerve cell and they hide in the nerve ganglia. Nerve ganglia are where all the nerve cell bodies are and where the nucleus of those nerves are. And reactivation of these latent infections occur in 1 in 3 people who have it, and then this results in shingles. So shingles usually causes just a rash, but it can also infect any part of the eye, and you can see that here in the top image. But it does most often cause damage to the cornea, and the most common problem with that is that it causes desensitization, so you lose feeling in your cornea. So if you were to see a scratch or something like that, you would have no idea, and that could lead to serious eye infection and damage. And so that would be like a common problem for people with contacts and stuff like that. And then an example of what could occur is corneal clouding, which is depicted here at the bottom. So working with VZV is very difficult, and the Kinchington lab is actually one of the very few labs that does study its biology. And one of the reasons it's so hard to work with is because its infectivity is also associated. So the virus sticks to the outside of the cell rather than detaching like most viruses do. And VZV is also very human specific. So there's no animal model we could put VZV into and observe what happens during latency. And latency is the period where the virus is dormant, not active, and it's not replicating at all. So the goal of this project was to help answer what happens during latency in these nerve ganglia. Because if we know what happens and we know it's made, then we might be able to target VZV to stop its reactivation and to stop shingles and its symptoms. But one of the things we do know is that during this period of latency, it produces an RNA called VLT, and you can see it depicted here as the full transcript, and then it's just the exons. And here's a diagram of it as well that will be used in the next slide. So it runs opposite of the VZV's genome, and it starts just before gene RF61. And this gene encodes for a protein that we know prepares the cell for infection. So since it runs opposite of this, it's believed that VOT is responsible for stopping it from being activated, which is why it's produced during latency. So the goal of this project was to produce virus without VOT. And the first step to doing that was PCR. So PCR polymerization reaction involves simply making more DNA. So first you denature it, which separates the two strands, and then primers are annealed onto it. And then these primers tell the polymerase where to start replicating. And so we used 5 different primers, 3 forward and 2 reversal. So all the forward primers start at different positions, and reversal 1 primer just takes out the oh my goodness.
Promoter.
Yeah, promoter region of VOT, but R2 takes out part of VOT itself. And so here in this diagram, it shows which part where in VOT it's taking out, but these primers don't actually take out VOT. What is happening during this PCR is that it's amplifying antibiotic cassettes. And what's interesting about these primers is that they have additional base pairs on the ends, which result in them creating 2 new strands that are attached to the antibiotic cassettes. And these are important for gene recombinating. So just taking out the VOT would disrupt the entire genome. And so by replacing it with something else, it allows it to remain stable. So we inserted antibiotic cassettes to replace VOT. And the 2 we used were kanamycin and ampicillin. And so with these PCR fragments of the kanamycin cassettes and ampicillin cassettes, we put that and the rest of the VZV genome into E. coli. And then the recombinating happens inside of it, and the antibiotic cassettes replace the VOT. And so the antibiotic cassettes also give resistance based on whatever cassette they are. So for the kanamycin inserts, those bacteria were plated on plates with kanamycin antibiotics. So that ensures they're the only bacteria that grow, and that's the DNA we're getting back out of the bacteria. But to test to make sure the replacement actually worked inside the bacteria, we use a restriction digest to perform cut tests. And so here is an image of my AMP DNAs after restriction digest with Hin-3. So here in the last well, you can see that this parent DNA has 2 bands really close together. But what you see in most of these samples is that the first band starts to move upwards, and that means that it was cut by the enzyme. And so the insertion did work. And while it's just shown using Hin-3, we also use the enzyme KPN1 and PST1. And so after we purify the back DNA back out of the E. coli, we transfect the DNA into HTRPE cell cultures, which just means putting them into it. And it works by putting them in a lipid that fuses with the cell membrane of the cells and inserts the viral DNA into them. And so they grow in culture for about 2 weeks, and we just want to see if virus forms. So these aren't from my own projects, but when you image the virus, you can tell growth by how much white space there is. And you can see in these day 0 pictures that as you go to day 6, there's a lot more white space, which means virus is present. So currently we have 1 recombinant virus growing and 6 more that are recently transfected, and we're just waiting to see if virus grows from them. And so once we have virus, the goal is to test if they go latent in human neuron cultures and if they do reactivate in these cultures, and just overall observe what happens to the VZV's growth if VOT is not present. And I really want to thank Mike again for all of his help over the summer, especially in the beginning, because I had to ask like 100 questions a day, and just to know what I was doing, and use a lot of help. And Kip as well for designing this project. and he was a lot of help, especially these last few weeks with, making this presentation, my abstract. And of course, David and Solomon, as well as Dr. Chen for just making Hillman possible and for this opportunity. So yeah.
Marcus, this is Lonnie Snyder. I'm with the Eye and Ear Foundation. Wonderful presentation. Dr. Kinchington said that you had a chance to, or you chose to have a real hands-on experience in his lab. What were some of the things you got to do in his lab in terms of experiences? It sounds like you got a chance to do quite a bit.
Yeah, I did a lot of cell cultures, growing and plating bacteria, transforming bacteria, imaging and running gels, a lot of that stuff.
Wonderful.
So I just learned online how to do screenshot from my Mac. So I just changed from Windows to Mac. So it's a learning curve for me. But Marcus, would you like to show up your face again and select one of your favorites slide?
Yeah.
Marcus, I cannot see you on my screen.
If you speak,
Speak up.
Marcus, you should
Yeah,
be able
speak
to
up.
get it.
Okay. Can you see me now?
Okay, got you.
Okay.
Thank you. All right. Great work.
Thank you, Marcus.
Thank you, Marcus, and thank you, Kip.
Thank you.
So our last speaker is Daniel Isak, and this is a special participant who is an undergrad at Pitt. And Daniel's mentor is Dr. Chris Donnelly. And is Dr. Donnelly here?
Yeah, I'm here.
Okay, great.
Should I introduce?
Yes.
Okay, great. So apologies, I had a prior engagement before this, so thank you for inviting me. And just want to say it's a pleasure and honor to welcome and introduce Daniel. Daniel worked in our lab briefly prior to COVID-19, and he showed a lot of motivation and enthusiasm for some of the work, and always very willing to try new and exciting kind of techniques and learn technical skills that I think are well beyond sort of the level for which he came into the lab. So when he asked to join this summer, to work in the lab this summer, we were very excited, and we tried to give him a little bit of an independent project where he could kind of run a little bit on his own with the help of a graduate student named Marilyn Nago, who is studying genetic factors that contribute to neurodegenerative diseases, in particularly ALS, FTD, which is ALS and myotrophic lateral sclerosis, FTD, frontotemporal dementia, and Alzheimer's disease. And so Daniel will tell you a little bit about the work, but even in the short time here, Daniel started working on a new gene that we're interested in, and we'll hope that he continues working with us throughout his undergraduate experience. Yeah, so I'll let him take over.
Thank you, Dr. Jhani, for the introduction. I appreciate it. So show my screen and get right into it. So I was for the Donnelly Lab this year, Department of Neurobiology, and we studied ALS. Dr. Chris Donnelly and Marilyn Nagu were my mentors. And my project this year was establishing the relationship between KPB3 and TDP43 localization. So to start off big picture, why is this even important? Why am I studying this? So obviously this is an ALS lab, and as you can see from the picture from the left to the right, in a little more than 4 years, Neil became someone who could barely even walk by himself. And even in 2021, we still haven't figured out a cure to this disease. And the Donnelly Lab hopes about that slowly and surely every step of the way. And ALS is a neurodegenerative disease, meaning it's the central nervous system slowly breaks down, which contributes to the paralysis symptoms of ALS. So as you can see on the graph on the left, on the red line, as neurodegeneration and the blue line, TDP-43, are extremely correlational. As one decreases, the other decreases, one increases, the other increases. So it's thought that neurodegeneration and TDP-43 have very intertwined roles correlationally. And as you can see on the right, TDP-43 is thought to be 97% at least correlational to ALS. So now, what does TDP-43 do? We know it has some sort of neuropathology, but how does it cause this? Well, TDP-43 gets mislocalized in ALS and FTD. So as you can see from the healthy cells, you can see that TDP-43 is equally distributed throughout the cell and isn't stayed in one place as compared to the unhealthy cell. As you can see, it's mislocalized in clumps in the cytoplasm, which creates this protein inclusion, which again is thought to create neurodegeneration and ultimately ALS in this instance. And so, based off the previous literature, in 2013, Dr. Koffler completed a GWAS study, a genome-wide association study between this TDP-43 mislocalization pathology with patients with Alzheimer's. And a GWAS study aims to associate genetic variants, SNPs, single nucleotide polymorphisms, with this TDP-43 pathology by genoming the everyone's DNA and running association correlation assays. And TDP-43 pathology is suggested to create this protein inclusions that all overall create neurodegeneration. So the results of Dr. Koffler's study is the most significant hit was called SCYL3. And that is a gene that codes for this SNP, this genetic variation, which is thought to highly correlate with TDP-43 pathology and therefore neurodegeneration. However, another very significant hit for KEFI-P3 that codes for another SNP. It was a very significant hit, and the distance between both these genes were a little less than 6 million base pairs. And speaking relatively, that's not very a lot of space between them. And so we know due to this linkage disequilibrium is this non-random assortment of different alleles due to close proximity. So one is more likely to be, during random assortment, that these two are more likely to be together than genes that are farther away. So a little bit about KEFI-P3. So it's codes for a Kinchington associated protein 3 called CAP3. And it's just to be a subunit which contributes to the Kinchington 2 motor protein. And that superfamily in general plays a role in the interaction with ATPase motor proteins, which play a role in transportation within the cell. And the hard part about my project this year is that KEFI-P3 is a very understudied gene. The literature is very basic. Not that many people have done a lot of bench work with this gene. So my work this year is setting the foundational parts for KEFI-P3 and its role in TDP-43 neurodegeneration and ALS. So my hypothesis was if KEFI-P3 is knocked down, then TDP-43 localization is altered. And the three big questions I sought to answer was, where is KEFI-P3 in the cell? What is the relationship with TDP-43 and this pathology? And then how does this affect TDP-43? KEFI-P3 within the cell. We ran a basic assay using HE cells, human embryonic kidney cells. We ran immunofluorescent staining using spinning disk and focal microscopy. And on the left, in the red, is a picture of antibody stain for KEFI-P3 Yes, and then on the right is alpha tubulin, which is in the green, and that is obviously because it's a Keynesian that uses microtubules. We thought it would be a great idea to see if these two co-localize, and they did significantly co-localize. However, there's so much microtubule around the cell, it wasn't specific enough to really call it a co-localization. But based off of this, we concluded that the KPP3 antibody did indeed work, and it was a good antibody to go from there. So to really answer the question, where's KPP3 within the cell, we went to literature, and the literature suggested that KPP3 is highly associated with the endoplasmic reticulum. So we sought to answer this question and see if that was indeed true. And so on the left is a merged picture of all the immunofluorescent staining we did for every single channel. There's DAPI for the nucleus, there's phalloidin for the cytoplasm, and then there's KPP3 in the red, as you can see in the middle, and then there's the calnexin protein marker in the ER green. And on the very right, you can see this Pearson's correlational coefficient to see the co-localization, how significant it actually is. And it's about 0.81, that's about the average for this co-localization. And that is pretty significantly highly correlated. So we just affirmed that the literature did indeed work, and it is indeed found near the endoplasmic reticulum. So the big question, what is this relationship with TDP43? Well, again, on the left is antibody staining for KPP3, and the right is TDP43. And as you can see, the Pearson's correlation coefficient was about 0.87. That's, from all the other experiments we ran, that was the lowest we found, is at least 0.95, 0.94, very highly significantly correlated. So yeah, if one is very significantly correlated, that's a good assumption to move on to step 2. And so we tried to do an siRNA knockdown of the KPP3 PCR product. And as science goes, you run the protocols, and we saw that there was a slight knockdown. If you can see the leftmost well that isn't the ladder, you can see there's a slight difference in lower in the DNA of the band. However, all the way in the far right, there's a band. It should be a no reverse transcriptase, so there should be no band. However, there was a band, which informs there's some form of DNA contamination. So the knockdown may not fully stable, as we'll see later. So on the left is we used siRNAs to knock down KPP3 using within the cell culture, using these AGK cells. And on the right is supposed to be the knockdown with KPP3 to siRNAs. On the left is supposed to be the scramble or the control, where there should be little to no knockdown. However, as you can see, the difference between the two is very minute. There's very little. It doesn't seem as though the knockdown really did indeed work. As you'll see from the validation of the knockdown, we have the treatment on the x-axis and the mean KPP3 intensity on the y, and you can see that there is no statistically significant difference. It doesn't appear that the knockdown really did indeed work. However, I just thought this was an interesting graph. So there's nuclear TDP and there's cytoplasmic TDP. Cytoplasmic TDP is the mislocalized TDP that comes with neuropathology. And we have the scrambled control in the siRNA, which is the knockdown. And although there seems to be this sort of pattern where even if you knock down with the siRNA, the KPP3, it increases TDP. And although we are not fully certain how yet, it's nice to see at least there's a pattern between this gene and this pathology. So last last assay we ran with immunofluorescence. Again, on the left is your merged, the most KPP3, on the right is SCOY3. And that was a gene with the most significant hit in the GWAS study, which my mentor, Marilyn, is actually studying. And we just wanted to see, do these, even though we know these genes are close together, but do they co-localize together? Are they close? As you can see, they look decently co-localized. The Pearson coefficient was high, 0.85. So we just thought it was interesting that the genes are close together and that yet they do co-localize decently with each other. And however, again, my work this summer wasn't nearly successful as science goes, but again, laying down the foundation work for future directions to be run. So for the future directions, I'd optimize the siRNA knockdown protocol to let it be successful, and for the RNA extraction to show significant difference in the bands. And I'd also like to run overexpression assay. So if we siRNA, we knocked it down, what if we overexpress it? What would happen in TP43 pathology? Would there be more? Would there be less? Find significant patterns. And we'd like to optimize these. I would love to optimize these protocols for the iPSC neurons. And again, understand the mechanism for mislocalization of TDP43. That's not so much what the Donny lab focuses on, but that'd be a great future direction to go for. And to run functional assays in KPP3, which is the next step to see truly what does this do, because not that many people have studied it. And I'll just like to give thanks to Dr. David Boone, Solomon Lifshitz, Dr. Chris Donnelly, my mentor, and Marilyn McGew for another mentor, and the George Duke Foundation for giving me this internship and opportunity, and all the members of the Hillman Cancer Center Academy and the NIH for grant funding. Learned so much, and experience was really invaluable. Yeah, so thank you.
I have a science question for you. So has you or your lab or anybody else looked at any kind of neuron model for the KIFA P3 localization? Like, is it in axons? I don't know. I mean, you said you're doing iPS neuron optimization. I just wondered if there's any other types of neuron cells that somebody's looked at it.
That's a great question. In my lab, there is iPSC neuron models set up. However, for KPP3, the neurons are very finicky. They die very easily, stuff like that. So we wanted to use the HEK cells. They're more robust model. We want to use that first before we go into neurons, because there you can give us concrete evidence before we dive into something where very finicky, die very often. Yeah, there hasn't been much research regarding the neurons yet, but obviously that's once we get significant detail within the HEK cell model, that's whenever we will step through the the iPSC neuron model. Yeah.
So there's a couple of neuron models out there that are a lot simpler than iPS neurons. And it might be worth looking at some of those, like Loomis and, I think SY5Y, that are very neuron-like, much easier to culture. So just a thought, but that's cool.
Thank you. Yeah, I know my lab personally, I know they like to use iPSCs, but I definitely understand where you're coming from. So thank you for the suggestion.
Hi, Daniel. I also have a question. I'm really excited to see another lab doing protein folding related project, because my lab is also working on protein folding disease in the eye. So the question I have for colocalization, have you ever thought about an alternative method such as a conventional co-IP or things just to convince the proteins that you are interested are really interacting with each other?
You know, that's a great point, actually. I don't think I have fully thought about that. But I think using different protocols would definitely be a great method to go about forward to really prove this experimental approach of co-localization. Definitely think it's a great suggestion. Thank you.
Thank you, Daniel. So same thing, why don't you choose one of your favorite slides? I guess probably this slide is wonderful. And speak up so that I can take a screenshot for you.
Yeah, I love this slide. So much hard work going into getting those images. Many attempts, many fails as science goes, but
Okay, I got you.
Thank you.
So it seems all of our speakers has finished your talk. And I really want to thank everybody. Today, as the last day of the Hillman Academy, it is also the first year that ophthalmology joined the Hillman program and accommodate high school students in the summer program. We have totally 4 high school students and 1 undergrad student participating on the OPT site. And it's involving in 5 laboratories with 7 clinician and basic research mentors. And the Ear and Eye Foundation of Pittsburgh, including Lonnie and Carrie and Heather and Craig, has all put a lot of efforts in making this happen. This summer program is supported by the Hillman Foundation, and we thank the mentors for sharing your experience and patience and resource with the student during the summer. And we really thank the students for your participation and passion. And hopefully you have learned something in the summer, which will be helpful for your future growth. And yeah, that's it. Lonnie, please add something.
Thank you, students. It was all wonderful. It was wonderful to just learn about everything that you've been experiencing this summer. I can tell you've been working really hard, and it's great that you get a chance to be introduced to science for those of you, particularly the high school students who never had maybe a chance to understand what it's like to work in a lab that is doing this kind of work. I hope it inspired you. I hope it gives you an opportunity to think about how you may be able to apply what you've learned in other aspects of what you end up doing in your life. So I have a favor to ask. Could everybody come on screen for just a second so we can get a screenshot? I'd love to, just kind of have that for our inaugural presentation. I hope that when we do these in years to come, we can actually do them in person, because it'd be lovely to see all of you. And sorry I didn't ever get to see you in person. So so thank you. I think we just need Zyaire and we have Young's got 2 screens there. But
so do we want the students all showing up?
Yeah. I was going to have them all on their screen, because I have everybody in one big sort of Brady Bunch style
Sofi
boxes.
Adams, Zyaire Beasley, Venice Lin, Marcus Waller, and Daniel Isak. Please show up.
The only screen I don't
faces.
see is Zyaire right now, but maybe she can't find her. Zyaire, can you come on screen? Okay.
Maybe she stepped away.
She may have stepped away. So let's Craig, if you would, get a snapshot, and it'll be nice to share this with others and our supporters and so forth. So there she is. Oh, good. Okay, great. Thank you, everybody.
One more reminder is the at 2 o'clock, all the students, please be reminded there is a closing ceremony hosted by David Boone. So please attend that closing ceremonies. And mentors are also welcome. I will send a link after this meeting so that all of you will have it. And when I collect the screenshot, I will also send it to the mentors and students. And thank you all for your participation and wish everybody a good day.
Let me introduce to the faculty Chuck Broadbeck from our board who joined us. We, again, in years to come, I'd love to have more people from our community who've been supporting this get to learn a little bit more about the students and what they're doing. So thank you, Chuck, for coming.
It was impressive.
Okay, have a good day, everybody.
Thank you.
Thank you.